Hello to all the participants. I am going to introduce you to the TotemBioNet tool, a tool that is the result of a collaboration with my E3S team, in particular Gilles Bernot, Hélène Colavizza, and Jean-Paul Comé. So what does TotemBioNet do? It automates the parameters identification for René Thomas discrete modeling framework. It is a formalism that models quality-state dynamic of biological regulatory networks. But how it does it? Parameters identification is based on two principal formal methods: whole logic and model checking for temporal logic. TotemBioNet combines efficiently both to check whether a set of Thomas parameterization satisfies biological properties. But before I show you the processing flow and the tool in action, I'm going to quickly define the concept that the tool handles. First, René Thomas framework, then whole logic, and finally model checking for CTL. So let's start with the René Thomas formalism. It is divided into two distinct parts, the syntax one and the semantic one. The Thomas syntax can be summarized by the definition of a regulatory graph. It gathers knowledge on regulation between several variables. Each vertex, here in yellow, abstract typically genes, transcript, or proteins, and each edge is labeled by a so-called multiplex, in gray, that encode the nature of regulation between several variables. Here, for example, M1 multiplex encodes V1 self-activation, M2 the inhibition of V1 by V2, and M3 the activation of V2 by the dimer Y1 Y2. So each variable is associated with discrete thresholds. Here, V1 has two thresholds, 1 and 2. This integer means that the variable is in a sufficient quantity to influence one or several targets. So for example, when V1 reaches its threshold 1, he is able to activate itself. But when it reaches its threshold 2, he is both able to activate itself and activate the dimer Y2. From that, each variable has a discrete domain from which the exhaustive set of state of the system is built, and it corresponds to the Cartesian product of variables domain. Okay, so now what about the Thomas semantic? It can be summarized by the definition of the transition graph. Here is the transition graph. Each vertex is a state of the system, and edges are the transition between states. A transition is a change of state, but for one variable at a time. Transitions are built using discrete parameters, the K parameters. Each variable has as many parameters as is that resource combination, where a resource is a predecessor of a variable. For example, V1 has two predecessors, M1 and M2, so has 4 parameters because it has two resources. The same approach is done for all the variables. The value the parameters can take correspond to the value toward which the variable is attracted inside a state. So the number of values a variable can take is finite, so is the number of values that the parameter can take. Given these two last points, the number of parameterization equals this product of two exponential. The role of the TotemBioNet is to select among this parameterization those that satisfy biological properties. But how? Notably, using whole logic. So whole logic can use this kind of expression level curves to constrain parameters values. Once discretized, these curves are translated into a whole triple, just like this, where pre is the precondition and post the post condition. Pre and post are the variable states before and after the experiment. The path is the observation of the threshold crossings of the variables over the time. From a whole triple, whole logic constructs the constraint on the K parameters that make the path possible from the precondition. Here's an example of how. Dijkstra-backward strategy calculates the constraints step by step from the post condition, and the genetically modified whole logic had a constraint on the parameter values. For example, to observe B+, it is necessary that B equals 0 in the state Q1, and that the parameter of B is strictly higher than its current value in the state Q1, and its current value is 0. And then the constraint on the parameters is propagated to the precondition. This is how we obtain the weakest precondition named WP. Host logic also includes path quantifiers. The quantifier exist and for all, and it is used typically when there's uncertainty about event order. Here, the exist one expresses that both paths are possible, and here that both must be possible. The weakest precondition that is calculated when the exist quantifier is encountered is a disjunction. When the for is encountered, it is a conjunction. TotemBioNet automatically computes this weakest precondition from a given whole triple, and that way select parameterization that satisfies dynamic observation, just like this profile. I present you the TotemBioNet workflow. So the TotemBioNet input file is divided into two parts. First, the regulatory graph right here that summarizes all the Onetoma syntax about a biological system and its biological properties about the dynamic, here with a whole triple, but also with a formula in temporal logic. In a complementary way, the CTL temporal logic is well suited to encode properties about the dynamic of the system. That's why TotemBioNet uses a model checking algorithm for CTL in order to verify the temporal logic formula. So from a regulatory graph, the tool is able to enumerate exhaustively the set of parameterization, and for each of them, the tool is able to verify if the CTL formulas are satisfied. If it's the case, parameterization is stored inside a CSV output file. Here is the power of TotemBioNet. Both combining model checking with host logic in order to both verify whole triple and CTL formula. There are two benefits. First, the expressiveness of biological behavior is increased. And second, the enumerated parameterization can be dramatically reduced by the weakest precondition. When a whole triple is given as an input, the weakest precondition that is calculated by the tool can be either conjunctive, and in this case, it reduces the domain of the parameters, so it reduces the number of parameterization that are enumerated. If the weakest precondition is disjunctive, in this case, it is evaluating during the enumeration. For each parameterization, the triple is verified. If it's satisfied, the CTL formula is verified, and if it is satisfied, it is stored inside the output CSV file. So the output CSV file contains all the parameterization satisfying both whole triple and CTL formulas. So let's start the demo with the case study of the mammalian cell cycle. The cell cycle is a series of events that lead to DNA replication and mitosis of the cell. And here's the biological question: how the progression of the cell cycle is controlled? To answer that, a regulation graph right here has been designed to answer this question, and it is composed of 4 variables: SK, A, and B, the main cyclin CDK complexes involved in the cell cycle progression, and EN and EP, their main inhibitors. Regulation between all these variables are encoded into this multiplex in the gray boxes. This regulation graph has been designed using YQD software, and TotemBioNet is able to convert it into a SMB file, that is the input file of TotemBioNet. Using in command line TotemBioNet, you can convert it using the YQD option. And we can obtain a SMB file where variable and regulation are declared. To verify if the graph is compatible with the progression of the cell cycle, we decide to formalize this progression using the whole triple H in it. Its path is divided into 4 sequences. In blue it's G1, in red S, in gray G2, and in green it's M. In addition, we use a first CTL formula named phycyclic to encode that all the path of the models that starting from G1 always end by cycling to G1, what the triple actually doesn't allow to verify. From the billions of initial parameterization, the TotemBioNet enumerates only 676 models that satisfy the weakest precondition calculated from H-init. Of these 676 models, only 609 of them satisfy both H-init and phycyclic. Let's watch the TotemBioNet in action. Using the SMB file, we add the properties H-init and phycyclic, and after that, we call TotemBioNet in command line. We use the option CSV to store the OK model into a table. Then we can find the results, and everything is obtained in 6 seconds. If we open the CSV file, we can find the OK models. Each line is a parameterization. Imagine that a biologist asks us, okay, is the order of events is important within a phase? And to try to answer him, we use the for all quantifier, just like here. And this for all quantifier in the whole triple hash perm allows to verify if all events orders are possible within each phase. And the answer is no. Why no? Because no model verifies the whole triple hash perm because the computed weakest precondition contains a contradiction. So the answer can be only no. After investigation, the biologist tells us that indeed S necessarily starts with A+, and N has a fixed order of its events. So we test this hypothesis with the whole triple H paying G1. And TotemBioNet returns in 2 seconds only that, yes, there is 240 models parameterization that satisfy both H-perm G1 and phycyclic. Now, having a better idea of what's going on inside each phase, we absolutely need to verify checkpoints of the cell cycle. So what is a checkpoint? It is between 2 phases, for example, G1 in blue and S in red, and a checkpoint ensures that none of the possible transition of S, here A+, must arrive before the completion of each transition of G1, here in blue. So here is the equivalent of the A+ transition. It mustn't arrive before the end of each event of G1 in blue. So this formula encodes that none of those paths that authorize the event A+ during the G1 phase must arrive. So the checkpoint G2M is formalized in the same way, and using TotemBioNet, we get 28 models of parameterization out of the billion that satisfy both G1S and G2M in less than 3 seconds. So now let's make a point about the TotemBioNet tool. I remind that it automates the parameters identification for Nettoma formalism. To do that, it uses 2 formal methods. On the one hand, the whole logic, and on the other, the CTL combined with model checking. The main purpose of the tool is to help the modeler to formalize biological knowledge, just like the cell cycle itself and its checkpoints, and to quickly verify them. You can find the source repository at this link. And about 2 improvements. First, we would like to allow the incremental analysis of parameterization by adding parameterization into the input of the tool. And the second one is to add a graphical interface to the tool using a Jupyter Notebook. Thank you all for your attention.